All right, this is going to also be a vision test. So I am Will Thrift from Genentech's Computational Sciences Group, and I'm really excited to be talking about equity, which I think is such an important topic with really practical implications. And today I'm going to be talking about equity at the interface of personalized cancer vaccines and deep machine learning. So personalized cancer vaccines, super exciting cancer immunotherapy where we really try to use the normal immune response that destroys cancer cells for our immunotherapy. And so the way that, generally the immune system tries to destroy cancer cells is through this pathway involving MHC. So MHC, it's a protein. It decorates the surface of the cells in your body, and it's responsible for presenting peptides. And these peptides are derived from the proteins that the cell produces. And so cancer cells, right, they're going to produce lots of mutant proteins, and so they'll produce mutant peptides. We call these mutant peptides neoantigens. And so neoantigens hopefully will get presented by MHC, and then a T cell will come along with its T cell receptor and hopefully recognize this neoantigen as foreign and destroy the cancer cell. Now, for whatever reason, if you have real cancer and you come into the clinic, it has set up some barricades to prevent the immune system from going through with this. But the idea behind a cancer vaccine is to sort of give these neoantigens in the form of a vaccine to kick the immune system back into gear and destroy the tumor. And what's pretty cool about this is there will be lots of neoantigens that any person has. They'll be unique to them. And then there's only a finite amount of space in the vaccine. So it turns into this ranking problem of like, given a patient's MHC alleles and given their neoantigens, which ones should we put in the vaccine that we give the patient? And so this sort of hinges on the MHC protein. So it's super polymorphic protein. There's more than 10,000 that have been identified. Each person will have 3 genes that encode for this, HLA, A, B, and C. So you can have up to 6 alleles. And this ends up being like a sequence problem of given an MHC sequence and given a peptide sequence, will it be presented? There's also the problem of T cell recognition, but getting that data for immunogenicity is very expensive, too expensive for training a deep learning model. So we just focus on MHC presentation here. And speaking of data generation, so it's pretty not well suited for deep learning because you can isolate the peptides that have been presented by MHC and read them out with mass spec. So, one experiment will generate thousands of peptides. People have done a lot of experiments. For part of this work, we collected all the publicly available data, and this ended up being a huge, 500,000 peptide allele pair data set. So really great for deep learning. There's this sort of issue where you don't see non-presented peptides, but I'll show how we can get over that. The issue, though, and why I'm talking to equity today, is that researchers who were collecting this data were just taking whatever samples they can get, which is totally reasonable. But it turns out that people who give samples to universities are not representative of people in the world. And I work for a pharmaceutical company, and we make medicine for people in the world. And so that is an issue for us. So if you look, people of European descent, super well covered by this data set. People of South and Central American descent, not so well covered by this data set. And so it's a problem of, can we improve coverage to these underrepresented ancestries? And so that's what I'll be talking about today. So in this work, we produced our own MHC peptide presentation model, which we call HL Apollo. It's based on transformers. I won't talk too much about it. Just a quick plug. We had this really nice way of dealing with false negatives in our data set that enabled us to get really state-of-the-art results. And so that's why I'm considering it, not the existing models. But what we care about for this problem is really the zero-shot allele generalization, of our model. So if you have an allele that's not in your training data set, how well does your model perform? Because a lot of these alleles from the underrepresented ancestries are not going to be in your training data set. So we decided to study this really systematically, where we just remove an allele from our training data set, retrain the model, see how it performed, and we produce this graph over on the right here, where you can see how much the performance dropped. And so looking, most alleles actually don't drop in performance too much. Like the median drop was about 10%, still much better than the other models we compared to that were in the literature, and those were trained on those alleles. So for the most part, it's got very good zero-shot allele generalization. And I think that deep machine learning models in general have pretty good zero-shot generalization if you have a good enough data set. The issue, though, is there's a lot of alleles where the zero-shot generalization is not so good. And if we're making a vaccine for a patient, we need to know if that's an allele that the model is not performing well on or performing well on, because we wouldn't want to use an allele that the model doesn't understand in ranking neoantigens. So we need to find a way to determine this. And so you can think of a lot of factors that would impact zero-shot generalization. So most obviously, I think, how similar is this new allele to existing alleles in the training data set? How well did you capture that allele? And then there's a lot of interesting factors that you can create from the data itself, from the peptides that allele presents. So we call the pattern of residues that are in a peptide that an allele presents the motif. How complicated is that motif is going to really impact zero-shot generalization. How similar is that motif to just sampling random peptides? How likely is an allele to present a random peptide? All of these, if you put them into just a simple multilinear regression, can really capture what the zero-shot performance will be on that allele. The issue with this, right, is that most of these factors are based on data that you would not have in the zero-shot setting. And so we need to do this in a totally a priori way. And the a priori factor that I talked about before was sequence similarity. And so if we just plot sequence similarity to performance drop, we see some correlation. The more different the sequence, the more your performance drops. But it doesn't really capture it well enough for practical use. And so we have to develop more factors. So for this, we were sort of inspired by an existing strategy in the field for model interpretation, which is making data motifs. So on the bottom here, we see making model motifs. On the bottom here, we see real data motifs. You just go look at the peptides, see how frequently you see different residues at different positions, and you make that motif. And so people have wanted for a long time to know how well does their model really capture these motifs. And so what they'll do is they'll get lots and lots of random peptides, send them through the model, see how well, or see which ones were scored very highly, and use that as the canonical peptides for that allele. And so doing this in this zero-shot setting, you get the motifs at the top, and you can see that regardless of how similar the MHC distance was, so left was close, right was far, you still generate good motifs. And this sort of shows us that we don't need real data. We can just use what the model thinks and generate all of our factors with that. And doing that, we can rebuild the multilinear regression used before, but now in a totally a priori way. And through this, we can capture with reasonable precision the predicted performance. And so now it just becomes a game of let's choose a threshold where we will use it. This ends up being pretty arbitrary. The worst performing allele in the regular non-zero shot setting was 69%, so we don't want to do worse than that. And then there's a spread, in this regression model, so we can add a standard deviation and get a threshold of 77%. So if the model here predicts that our performance should be over 77%, we'll use it. And so we can see that including this, we can really greatly increase the full genotype coverage of our ancestries. For people of South and Central American ancestry, we're achieving more than 10% increase in the genotypic coverage, which I think is pretty good. It's sort of like a nice little story, because usually when you talk about data set bias in deep learning, it's not a nice story. It's a very negative one. But here, it was really deep learning that enabled this zero-shot generalization, and that enabled us to cover these underrepresented ancestries better. But that's not all we can do. You know, now that we can evaluate even alleles that aren't in our data, how well the model will perform, we can choose alleles that will improve our coverage the most. And so you can write just like a stupid, simple algorithm to see which alleles or combination of alleles will most increase coverage. And so I think a 90% full genotype coverage of all those ancestries shown before should be very possible. And it turns out that in just 9 alleles, you can achieve this, which is great, because any large biotech company with a good experimental group, could really collect all this data. And so doing so would really improve clinical trial equity. So that's my talk. I hope that you enjoyed it, and I'll take any questions.
Thank you very much for the talk. So I was wondering, when you looked at the sequence similarity between the MHCs and the performance drop, so did you there just look at all positions, or just the positions in the binding cleft, or which are close to the binding cleft? Or?
Great question. So here we're using the NetMHC-PAN pseudosequence, so just what's in the binding cleft. I have seen some issues with the NetMHC-PAN binding cleft. If you're interested in that, actually, I'm giving a talk tomorrow on MHC class 2, which I want to plug real quick at like 1:20 or something, and I'll dig into that a little bit. But yeah, ideally, these are just residues that are in the binding cleft.
So great talk. I think in your example, you showed an improvement of 10%.
If you're trained.
Yeah, indeed. Do you think it's enough?
I mean, so not to dunk on my competitors, but 10% in this plot is a smaller gap than going from HL Apollo to mixed MHC-Pred or NetMHC-PAN. So most people use NetMHC-PAN. It's a fantastic model. It works really well. So I think that for me, it's good enough.
Any other questions? I think we have time for one more.
I was just wondering if you know about the all of us data set that is being developed in the United States. It's precisely to address inequity in the distribution of demographics that are normally analyzed. And if you don't, do you think you could use that kind of data set to test your prediction?
So I am not familiar with it, and one of the reasons I was excited to talk at this panel is I'm hoping that people will tell me about what else is being done in equity and how we can expand that, because I think it's something that we have the resources to really address. And so I would love to talk to you after this about that data set, because I think this sort of research, it's not just important from like an ethical standpoint, and it absolutely is, but it also has like serious practical implications. If you're making an inequitable model, you are making a bad drug, and that's a problem. So I'd love to talk more. And if anyone else has any ideas, let me know.